Thanks the organizers for giving me this opportunity. Actually, my student, Diwu, was supposed to give this talk, but he couldn't get a visa, so I'm giving this, and I only realized 5 minutes ago that I have 10 minutes, not 20, so you're gonna see stress time. See how it goes. Okay, so we're gonna talk about both tasks, predictions, and designs, but first we need to explain what RNA splicing is. So in general, we have DNA transcribed to RNA and then translated if it's coding for proteins. We care about alternative splicing where the exons, which are those rectangle, colorful rectangles, are spliced together to produce different isoforms that can be translated. Like this exon can be included or excluded, leading to different isoforms, and they might have different functions, lead to disease, etc. The way we typically quantify this is we have junction spanning reads in RNA sequencing that can quantify this red exon inclusion or exclusion. And if you think of the entire transcriptome, that's going to be our input data for our deep learning or machine learning models, quantifying it across many conditions. There's a lot of splicing regulation going on between different, to regulate those events. There are core splicing machineries with splice sites, branch point, polypyrimidine tracks, like shown here, and they're very, generally very close, few hundred bases away from the exons or in the proximal introns and various RNA binding proteins that again bind in this proximal region, so relatively close. And you're gonna believe me that RNA splicing is really important, otherwise we can talk about it later, and that we really wanna predict it. And the way it's done, I'll divide to two main ways for the sake of this talk. Local informed models. They look at this red exon, the flanking introns, the flanking exons, and they extract features from those 300 bases or so flanking regions. These can be predefined, manually created features as shown here, or you can learn them with CNN filters, etc. You stick them into a new learning algorithm, maybe with the specific tissue type, and you predict exon inclusion or exclusion. And this was done in 2010. That was the first time it was done. And then that was decision trees, then Bayesian neural networks, then deep learning with manual features or CNNs, etc. Now, the other approach was started with Splice AI, which is a very successful tool for predicting cryptic splicing. So it looks at very large windows, 10 kb, around a specific position and tries to predict is that going to be a splice site, yes or no, and whether it's going to be affected by mutations. Has found very useful for clinical diagnostics of cryptic splicing. And then that was extended for tissue-specific splicing in pangolin in 2020. And now there's this, I guess, trend for larger and larger models like Informer, looking at over 110 kb. And there's more and more of those foundation models, and I'm sorry if your favorite one is not listed here. So is bigger really better? So if you think of splicing, here's an example of a 160 kb gene, and this is 7.5 kb alternative splicing event, which is neuronal specific. And you can see the exons are tiny, and those regulatory elements that I told you are in close proximity, but we're learning those huge models over everything. So the question is that useful or not? And so we want to be able to use large language models, but maybe exploit the known annotation and the known biology around it. And we want this to be very tissue-specific, accurate, and be able to also achieve state-of-the-art predictions, tell us where the regulatory elements exist, and maybe then design and correct splicing for therapeutics. So here is our proposal to how to do this. So we first pre-train BERT transformers on splice sites across the human genome. And once we've had that done, we can develop this model. Now you can see 4 different transformers, but this time they're sitting across those splice sites that we know from the annotation where they exist. And we can combine them together through an MLP with extra features that we can extract easily. And then we can predict both inclusion levels and changes in inclusion levels, which are called psi and delta psi shown here. And so here is an example of how it can improve in predictions for tissue-specific splicing and splicing in general. This is splicing performance on 3 tissues in GTEx. This is pangolin improving this. Obviously, it's tissue-specific splicing, and this is Trasper. Now, this is all splicing events. Of course, they're generally either highly included or excluded. But if you look at specifically changing events, then you can see, of course, performance degrades, but the improvements are much more significant. And I could show you this for differential splicing calling, so saying when it's changing or not changing. This is, again, pangolin, which is current state of the art. This is our model. This is AUCs. But of course, these are small sets, so you should be looking at AUPRCs. Of course, the numbers drop, but you can see a very significant improvement in prediction power. We can also generalize to unseen conditions. The way we do this is we use an encoder that encodes the latent space representation for every cell type or tissue. And then if we try to use GTEx to predict cell lines and encode, that's what we get. If we use this, we can improve it further. We can also predict other types, not cassette types. This is 3' and 5' predictions. This is pangolin. This is us. Also say that we seem to be performing and improving significantly. And we also learn splicing regulatory elements. This is what happens if you try to take weak splicing and improve it, or do it the other way, and you can see the effect because you're basically replacing weak splice sites with strong ones or vice versa. So that's just a sanity check that splice sites are being learned and do what they do. This is for condition-specific regulatory elements that we map through ENCODE, and we're generally getting them in the right direction most of the time. Of course, it's statistically significant, but there's room for improvement. And these are validations. This is done with ASO. This is also an SQTL for a specific disease. I don't have time to talk about this. We did this with ASOs, and this is with DCAS13 targeting for another gene. If you go mutagenesis analysis for a lot of variants, this was done for CD19 exon 2. A lot of variants, you can see splice AI performance here and Trasper. So significant improvements there as well. We want to move to generation. So how do we do this? We use Trasper as our oracle or our teacher, and then we feed it into a Bayesian optimization models called BOSS. This is also an encoder decoder transformer, but it learns a latent space representation and updates it iteratively under the constraints that the user give for the optimization function. Again, I don't have time to go through the details. We can talk about it later. Here is an example of what the user wants. They want to increase inclusion for lowly included exons, but no more than 30 positions that would be mutated. And as you can see, it focuses on the splice sites, makes sense, but it also hits areas in the introns that we saw overlap RBP or produce RBP motifs that we can recognize in many of those cases. Here's another one. This is DEM1 exon 16. This is neuronal specific exon. And we're targeting areas that we know from mutagenesis analysis have regulatory elements, and you can see that they concentrate there, which is very nice. And this is just to check the efficiency of BOSS. So what you're seeing here is we say, please increase inclusion, at least 20%, no more than 30 positions. And we're comparing it to random mutations or for a genetic algorithm that were proposed by another algorithm for a related task. And you can see for the green and the blue that we are way more efficient in producing samples that actually meet the criteria that the user wants, and that the maximum performing sequence generated is also better performing. So where does it take us in terms of future directions? I would say everything that I said that we're working, and I showed you examples of how we're improving, are also future directions. I think there's still a lot of room to improve. I don't think the work is done here. And I would encourage you to engage. I think it's an exciting area. And I'll just finish by thanking the people who did all the hard work. Dee led this large effort with a lot of support from Sun, our developer who supported all this work. Benjamin did the experimental essays that I showed you. Jake and Natalie are our collaborators who worked with us on developing the BOSS algorithm Peter commented on the experiments, and Anupama is sitting right there. She's now a postdoc, but she was previously in my lab and initiated all this effort. And this is all the lab and the funding, and thank you for your time.
Thank you very much. We have time for a couple of questions.
Hey, Joseph, cool talk. I wanted to ask if you could give us some insight. If I understood correctly, you basically use a smaller window around the exons where you want to predict now the splicing. But maybe I got it wrong compared to the other methods, like Splice AI. So why do you think you're doing better at the end? Do you have like a smaller model and you find a better optimum with this, or are there other tricks that you didn't have time to talk about?
Yeah, so in general, so in the paper, what we did an ablation study and we checked all the things, the component of the model that I sort of flashed. So the target function, the pre-training, the fact that we're doing an RNA pre-training and not like DNA BERT or something like that. All of those components increase our performance, basically, right? But I think, again, if you're training a huge model, then you're spending a lot of effort and compute power on things that are maybe not as relevant for the specific tasks that you're interested in.
Thanks.